Okay, now let's discuss about eubacteria. So eubacteria are the advanced form of bacteria. Initially we have discussed about archaebacteria, but eubacteria are the true bacteria. These are the true bacteria. The best example is Anabaena Nostoc. Let's discuss one by one. So they possess a rigid cell wall, a definite cell wall, and the cell wall is made up of peptido, that is glycan. The cell wall is made up of peptides, bonds, and glucose or glycans, that is polysaccharides. So it contains rigid cell wall. Many have, not all, many of them have flagella in the cell or in its structure. Let's move into the types of eubacteria. Cyanobacteria is one subgroup of eubacteria, then followed by mycoplasma is another subgroup. We also find actinomycetes also present and rickettsia also present. Let's discuss one by one, that is cyanobacteria. It is sometimes called as blue-green algae. These cyanobacteria are gram-negative photosynthetic bacteria and is the first bacteria to liberate oxygen. To liberate oxygen during photosynthesis means they contains both PS1 and also they contains PS2. So photolysis of water is found to present in this particular bacteria. So these are called as oxygenic photosynthetic bacteria because they liberate oxygen to the atmosphere. Initially, at the time of origin of life, the atmosphere was reducing atmosphere. But due to the evolution of this particular bacteria, the atmosphere regained the oxygen. So it is called as oxygenic atmosphere, and oxygen returns to the atmosphere through this bacteria. So cyanobacteria is sometimes called as the blue-green algae. The best example is Nostoc Anabaena. They are classified under Cyanophyceae, or you can classify it under Mixophyceae. Now, occurrence of cyanobacteria, the habitat of cyanobacteria, they are mostly freshwater, that is aquatic. Some are found to be present in seawater, marine. Some are found to be present in moist soil. And the cyanobacteria are also found to be present in high temperature water, or thermal water. And they also found mutual benefit or mutual association with other groups of plants like algae, like fungi, Bryophyta, Pteridophyta, or Gymnosperm. In cyanobacteria, the symbiotic association is seen, that is Anabaena with Azolla, or we find this cyanobacteria associates with the cycas root called to form coralloid roots, and sometimes fungi, algae are associated to produce lichen. And also we find this cyanobacteria present in some protista called as cyanelles. If cyanobacteria present as symbiotic association with protista, these are called as cyanelles. We do call them as cyanelle to this form of bacteria. Okay, so now move to the structure of the cyanobacteria. Mostly they are unicellular, single cell organisms. Some are present in colonial form. Some groups are present in colonial forms. Some are present in filamentous form. So the filamentous form is known as trichomes. The filamentous form is called as trichomes. So trichomes is the filamentous form. So let's discuss. This is the trichome present, and we find a mucilaginous sheath is found to be present. This mucilaginous sheath is present. Outside, this is the mucilaginous sheath present, and these individuals are prokaryotic cells. These are prokaryotic cells are found to be present, and this is called as trichomes, and it is a filamentous form, example Nostoc. And trichomes are surrounded by a sheath that is called as mucilaginous sheath. They lack flagella, they don't have flagella in the cell structure. So flagella also absent throughout the life cycle. So in the life cycle they don't find any flagella present, and these are mostly prokaryotic cells as the nuclear membrane is absent and membrane bond organelles are also absent. And also chromatin matrices are found to be present at the center. These are naked DNA present at the center, and the cells are surrounded by a mucilaginous sheath for protection. Outside covering present, a sheath is present outside the cell wall that provide protection to this particular wall organism, and peptidoglycan is present. This is the true cell wall composition present, or it is called as murine. So murines are found in the cell wall, that is peptide bonds are peptide and glucans are present, glycans are present, and their resemblance with the bacterial chromosome, as the chromosome is nucleate called. So the inner mass, the inner part, this one is called as the nucleoid. The circular chromosome or DNA is present, and we find the lamellosomes are present. These are the inclusion. In bacteria, normal bacteria, we found mesosomes present, but in eubacteria, BGA, we find lamellosomes present, which helps in attachment with this DNA during the time of binary fission. It helps in replications of DNA or formation of two daughter cells. And this is the mucilaginous sheath present, and this one is the cell wall present, and this is the cell membrane present, and we find these are the thylakoid membranes present with pigments present. and some cell inclusion present, like oil droplets present, gas vacuoles present, volutins present, etc. etc. So the protoplasm is divided into centroplasm and chromatoplasm. So it is divided into centroplasm and chromatoplasm. Means the outside part, the peripheral part, this peripheral part of is called as chromoplasm. Chromoplasm. Why it is called as chromoplasm? Because it is colored. Why it looks colored? Because the thylakoid membranes are present having pigments. But the inner mass is called as centroplasm. It is colorless. Centroplasm because the nuclear DNA is present. The nuclear DNA present. So this is what we find the protoplasm contains, that is centroplasm, and chromatoplasm, which is colored form, and this is colorless form. This is colorless form. Again, we find the centroplasm contains the nuclear materials and the chromatoplasm contains the photosynthetic materials, that is thylakoid, which are colored. Cell organelles, the membrane bound organelles are absent. Yes, they don't have mitochondria, they don't have chloroplast, they don't have, that is endoplasmic reticulum or Golgi body, etc. etc. They don't have all these things. But 70 is ribosome present because ribosome is the only cell organelle present in both prokaryotic cells and eukaryotic cells. They contain lamellar instead of mesosomes, because in normal bacteria mesosomes are present, but here we find cell inclusion, cell invagination or enfolding substance called as lamellosome. And this lamellosome gets attached with the nuclear membrane and helps in replication and helps in binary fission, formation of two new daughter individuals. Gas vacuoles are present that helps in floating to maintain buoyancy, which helps in floating in water. Photosynthetic pigments present in thylakoids are present in two different forms. One is chlorophyll A, another one is phycobilins. So phyco means these are the algae, bilins are the bile-like pigments present. Not the bile pigments, but the chemical composition is equivalent to the bile pigments or liver, so we do call them as bilins. Phyco means present in the algae. And the phycobilins are in the two form. One is phycocyanin, phycoerythrin. Phycocyanin produce blue color and phycoerythrin produce that is called as red color. So they contains blue color, they form body color. So phycobilins provide the body coloration, the body color. But chlorophyll helps in photosynthesis. They helps in photosynthesis. Got it? So chlorophyll helps in photosynthesis. Phycobilins provide body colorations. The reserve food materials are present in different forms. Cyanophycin granules, reserve proteins. Then beta granules, the reserve fat droplets. Then cyanophycin starch, the reserve starch as the reserve food materials. The starch as the reserve food materials. Volutin bodies are present to resolve the phosphate. Polyhedral bodies are present to resolve the rubisco. And the reserve food matters, those presence helps in metabolism, that is they do helps in synthesis, that is called as anabolism of the cell, and also they helps in catabolism of the cells, that is breakdown. So they helps anabolism and catabolism. And let's go to the Guy-Dukovas principles. So this cyanobacteria follow Guy-Dukovas principles, and Guy-Dukovas principles helps chromatic adaptations, mean colored adaptation. For example, suppose this is the water body present. If cyanobacteria present at the surface, it will imparts a different color. If present at the bottom of the benthic region, it will imparts a different color. So the surface, the wavelength of light imparts blue color, and in the deep, the wavelength of light imparts red color. So this two variation of color on the surface of water, the different wavelength of light absorption is called as Guy-Dukovas principle. So pigment coloration changes according to the change in the wavelength of light. So trichodesmium erythritum present in the benthic region show red color, whereas blue-green algae present on the surface show blue color. So this is about the chromatic adaptation. And let's briefly discuss about what the structure do contains. That is, they contains the gas vacuoles are present, circular chromosomes are present, that is circular DNA present, mucilaginous coverings are present, surrounding cells are present, they provide protections. And also in the metabolism we find that they are self-dependent organisms, but they are autotrophic. They are autotrophic. That means they produce photosynthesis. They use carbon dioxide to release food materials and liberate oxygen by means of photosynthesis. And also they liberate nitrogen, means they are nitrogen fixing bacteria. They fix nitrogen, converts to ammonia and increase the fertility of the soil. And this nitrogen fixation in cyanobacteria is done by a specialized cell called as Heterocyst. So nitrogen fixation is an anaerobic process, and it is done by a special bacteria cell called as Heterocyst. So these Heterocysts are present in the bacteria. These are the Heterocysts present, and these Heterocysts helps in nitrogen fixation because they contains the enzyme nitrogenase enzyme, and this nitrogenase enzyme only active in absence of oxygen. It is only active in the absence of oxygen. So as it is active only in the absence of oxygen, so it activates the nitrogenous enzyme and helps fixation of nitrogen. So the heterocyst are specialized cells where the nitrogen takes place, but nitrogen fixation requires the enzyme nitrogenase, and the nitrogen only works in the absence of oxygen, for which the heterocyst cell wall is very thick in nature. So it is an anaerobic nature for which the heterocyst nostoc, that is nostoc, the cell wall is very thick. Why? They prevent the entry of oxygen and activates the enzyme nitrogenase for nitrogen fixation. So heterocysts have terminal pores. The matured heterocyst, when the heterocyst becomes matured, they have pores opening to liberate nitrogen. And this matured opening also contains certain granules present, and these granules are called as polar granules are found to be present. That do helps in fragmentations and do helps in formation of new bodies, new asexual bodies for reproduction, that is called as hormogonia. So photosystem 2 is absent in heterocyst. This photosystem 2 helps in photolysis of water. Photolysis of water, a breakdown of water to release oxygen. So heterocyst never release oxygen because photosystem 2 pigment is absent. System is absent. So oxygen is never liberated by heterocyst. But in trichomes contains photosystem 1. So photosystem 1 is active in heterocyst, but photosystem 2 is active. It is active in trichomes. So it is active in trichomes. It is active in trichomes, but 1 is active in heterocyst, but 2 is active in trichomes. So PS1 generates ATP for nitrogen fixation. So this nitrogen fixation needs ATP, and to this ATP, this is used to fix nitrogen, and this nitrogen helps in increasing the soil fertility. Heterocyst also helps fragmentation. Suppose this is heterocyst. When the heterocyst matures, it comes out from the fragments. When it comes out from the trichomes, then each becomes fragments. Each are new fragments, and that develops into new individuals. And this heterocyst itself forms hormogonia. And these hormogonia are asexual bodies to give rise to new trichomes or new individuals. So nitrogen compounds release out when there is a breakage of these fragments. When the fragments breaks, the nitrogen compounds are released out, liberating nitrogen. So let's move to the reproduction. Cyanobacteria asexually. Binary fission is the most common method. Sexual method is totally absent, and multiple methods are present like hormogonia, that is binary fission present, and we also find rhizomatic division as we already discussed, and also we find fragmentation in filamentous form. It is unicellular form. If it is unicellular anabaena unicellular form, they go for binary fission. If it is filamentous form, they go for fragmentation. So this fragmentation, the leak fragments develops into new individual, and these fragments gives out, that is nitrogen is liberated to them, and these fragments forms new filaments. Heterocysts germinate under special condition, that is called as hormogonia, as I have discussed, and this hormogonia forms mucilage develops around it, and this hormogonia they produce mucilage, and simultaneously what happens inside the mucilage increases in nature. Inside the nucleus, new individuals are produced. This is how we find. And some dead heterocysts, the dead trichomes cells are called as necridia. So dead appearance of any cells is called as necridia. Akinitis. What is akinitis? In the trichomes, what happens? Certain cells accumulates reserve food materials and become thick in nature. And this reserve food materials thick in nature is called as akinitis. Under favorable condition, they comes out from the fragments, and these becomes new fragments in individual. These becomes fragments, and these new akinitis then develop into germinates into new individuals. So this is how akinitis also helps in asexual reproduction. So akinitis accumulates food materials and the germinates under favorable conditions and give rise to new. What are the importance of cyanobacteria? They liberate oxygen to the atmosphere as they are oxygenic photosynthetic bacteria. They help evolution of aerobic life. That is, initially the atmosphere was reducing. Later, by the presence of this cyanobacteria, atmosphere is ample with oxygen. So they help aerobic life to survive due to presence of oxygen. Increases cyanobacteria. Cyanobacteria and soil fertility. So it helps in increasing the nitrogen is converted into ammonium compounds, will increase the soil fertility, is increased due to the decay of this bacteria. The best example is Nostoc and Tolipothrix, which are used in rice fields. Anabaena is also used sometimes. Cyanobacteria show symbiotic association in plants. Nitrogen compounds supplied to the host cells and both partners are benefited. Spirulina is a cyanobacteria which is a single cell protein and that contains heavy deposits of protein materials present rich in proteins which are used as space food materials. Spacecraft use this as a protein supplements. And extracts of lignema, the extra lignia helps in antibiotic formations. Microcyst aurigonacea causes algal blooms. So these are the harmful aspects. Also, Anabaena floss aqua also cause blooms. Blooms causes toxins to the water materials. So toxins are produced which are harmful to the aquatic animals. So water sources become very harmful to these animals. Oxygen in the water goes on decreasing. Due to the presence of algal bloom, the oxygen decreases in the water. Aquatic animals may die. Aquatic animals may die. So this is all about, that is this is about cyanobacteria. So we have completed cyanobacteria structure, its life process, its uses, its disuses.